I think we can start now. So I'd just like to make a quick announcement for all students. So I think this will be the last seminar of this series for the fall term. And this will continue again in the winter term, beginning, I think, in January. I don't remember the exact date, but you'll be posted on that. So it is my great pleasure to introduce our invited speaker and final speaker of this term, Professor Francis Lin. So Professor Lin received his PhD in physics from the University of California. He then went on to complete his postdoc at Stanford University School of Medicine in 2008. He is currently a professor in the Department of Physics and Astronomy and holds a professorship in lab-on-chip technology at the University of Manitoba. He is an expert in the field of microfluidics with an extensive publication list of research articles and reviews. His main research focus is the application of interdisciplinary approaches to better understand immune cell trafficking in complex tissue environments and in developing microfluidic systems for biomedical application. So without further ado, please welcome Professor Lin.
Okay, thank you very much, Dr. Grant, for that nice introduction. Allow me also thank Dr. Well and the seminar organizer for inviting me to present my work. So as Dr. Grant mentioned earlier in the introduction, I'm in the field of lab-on-chip and microfluidic research. And in fact, we were chatting earlier, the largest microfluidics conference was held in Montreal about a month and a half ago, and we're in there. So there's lots of nice work we're able to learn from, and definitely over the last I think 2 decades or so, microfluidics has been heavily driving biological and biomedical applications. So today, I'd like to take the chance to share a bit of our own story, that is kind of our journey trying to make microfluidic devices a little bit easier for bio users for different assays, particularly for cell migration and trafficking research. That's kind of where the title of my talk is coming from. Using what we call the passive flow microfluidics, we're trying to make the biological assays easier for the end users. To start, I'd like to just try to move to the next slides. Oh, okay, go fast. Just a quick overview of my research program. We started about 16 years ago in Winnipeg, Manitoba, and as mentioned, our work is heavily involving lab-on-chip technologies. The core idea is to use those new technologies to get a better understanding of the mechanism for biological systems. And from there, that hopefully help us to enable different applications. And one of the focus is the translational medicine. And over the years, we have been working on 3 major areas. One of them is immune cell migration and trafficking. Second is to look at regenerative medicine. And over the last few years, there's a new direction that we're opening up, that we are developing new biodiagnostic methodologies. So here, this slide shows a few examples that we have developed over the years. For immune cell migration, one of the things we've been looking at is how T lymphocyte migrate and position in secondary lymphoid tissues. And we use microfluidic device to configure different simple coexisting chemical gradient to mimic the secondary lymphoid microenvironment. And there we use microscopy to visualize T cell migration in the simulate microenvironment. And that in combination of mathematical modeling and simulation, in there we observe a quite interesting repulsive migration of T cells in coexisting C cell 19 and 21 gradient, which is secreted in the T cell zone of the lymph node. And then the one below that is a more recent development of 3D skin on a chip model, and we use that to look at T cell transmigration as the involved co-culture of endothelium cells, epithelium cells, and then eventually T cells to look at their migratory responses. For regenerative medicine, the few examples I put in there, one of them is to look at the effect of electric field on directing skin epithelium cell migration. So we're able to pattern the skin tissue cells in different geometries. And here is example that we pattern into the square shape. Then we apply electric field to direct their migration. Let's see if this movie is playing. Okay, it's playing now. So the cathode is on the top, anode, the positive is in the bottom. As you can see, over the few hours experiment, its whole square is moving downward towards the positive side of the externally applied electrical field. So in fact, this electrical curve was able to maintain the integrity of the cell cluster geometry. So the whole cluster is moving directionally. And the one below is another example. We are looking at the migration of adipose-derived stem cells. So using a sequence of patterning technologies using the micro devices, we're able to force pattern the stem cells on one side of the microfluidic channel. And then we apply a boost factor gradient to induce chemotaxis to allow the cells to migrate toward the higher concentration side of the channel. Then we apply a stack gradient of trypsin EDTA so we can isolate those highly migratory cells. And then after that, the cells behind, which is we believe is a low motility cell, can also be extracted. And from there, we can compare their migratory motilities later on. So this kind of speaks to the future potential of purifying highly migratory stem cells for different biomedical applications. Then finally, in more recent years, we have been developing lab-on-chip-based point-of-care diagnostic method for different diseases. One of them is on the left is we develop our own on-chip method, so we can directly start a small drop of blood, isolate the immune cells from there, and then on the same device, we're able to perform cell migration assay. So the vision there is to eventually, this simple method can be adapted in the clinic, so we can use this what we call the cell functional biomarker for disease evaluations. And the one in the middle and on the right are some development to target chronic kidney disease. The one in the middle is a simple paper-based micro device. We use that to quantify C-reactive protein in the blood as a biomarker for CKD evaluation. And on the right is another chip that we developed has a higher throughput. We're able to quantify albumin from the urine samples of the CKD patient. So I spent some time at the beginning, kind of give you a general idea of our direction, but then with the time we have today, so I pick just a few examples focusing on cell migration and trafficking. I thought there's a more clear storyline in there, kind of share our journey over the years to make this cell migration easier and simpler for the users for their applications. Okay, so there's a lot of environmental guiding factors can direct the migration of different cells. The one on the left is example from our experiment showing human neutrophil chemotaxis. We can maintain a stable chemokine concentration gradient then over about 20 to half hour you see this human neutrophils clearly migrating toward the higher concentration. So the green color showing the concentration of the gradient. I put a special arrow in there. That's the cell you can actually see quite interesting. Has a very slow start. But once it's activated, when it sees the green, they were able to in the end overtake lots of the early starters. So it's kind of interesting observation to us. And then I will repeat on the right, is the example of with the electrotaxis. So applying external electro field, we saw this whole cluster pattern in a square shape of skin keratinocyte are moving down toward the positive side of the EF. All right, so there are lots of in vitro cell migration assays that people has been using to study the migration of different cells. Transfer assays is probably one of the most commonly used migration tools. And there are a few others, Sigma chamber, Andros assay, and there's a micropipette assay. I won't have time to go to the details of each assay, but essentially, for all those assays, you basically put in your reagent into a chamber, let it diffuse. There is general gradient over time, so you can observe the macro response of the cells to the gradient. Well, it's widely used. One of the common issue of those conventional migration assays is the lacking of the accurate gradient controls. The gradient shape change significantly over time, as shown in the plot on the bottom right of its slides. Eventually all disappear. So it's hard to quantify is this a cell really responding to the gradient, or eventually in the end of his experiment, there's simply no gradient is present, and what is causing the cell response at that time point. So over the last 20, 25 years or so, there's a strong trend in developing lab-on-chip method for cell migration assays. YSS group in Harvard is the first to demonstrate the use of microfluidic devices for studying immune cell chemotaxis. Nuri Zhang, who's my PhD advisor, who has now moved back to South Korea, continued his research when he started his lab at UC Irvine, which I did my PhD with him in the lab. So here's a picture of this quite well-known called Christmas tree design of the micro device in PDMS. So you can inject buffer and chemo tracking into this network of microchannels to generate a stable and well-defined profile of chemical concentration gradient. At the time when I did graduate study, I also did something to configure dynamically change chemical gradient. So here is a movie showing the switch of gradient directions dynamically in the same microfluidic devices. Another thing you can do with this kind of synthesis, you can configure coexisting gradient of different chemoattractant. The movie showing there is, again, human neutrophils in a competing gradient of two different chemoattractant. And we ask how the cell will make a decision when you have two gradient along the opposite direction. And then more recently, from a simple cell-based micro device, there's a trend to develop more complicated cell culture-based 3D with the organ-on-chip model to better mimic the tissue environment. And that really people has a vision to help to reduce the use of animal models. The one down in there is actually from my earlier work that we are looking at electrotaxis of skin T cells using intravitreal microscopy from mouse. One day, hopefully, those organ-on-chip model can really closely mimic the tissue environment, and hopefully at the time we don't have to use as many of those animals for the cell migration studies. So it's kind of the vision in there. All right, so from there, I'd like to kind of start to talk about our journey over the last, 10 plus years, trying to make those devices easier. And the issue I hopefully can elaborate in these slides. This is a picture from about 25 years ago when I first started. We have a very small microfluidic devices shown in there, about the size of a credit card. But you do require a very large specialized facility to go with that. That speaks to you need multiple syringe pumps using tubings to connect the chip to allow this flow-based reading generation. You need a large microscopy setup so you can visualize cell migration in real time. At the time, we are still using VCR and monitor to record the cell images. So later on, we have to convert the videotape into digital images into the computer, then do cell tracking in there. And to control the temperature, CO2 injection, humidity, you also have those enclosures to go with that. So this is a big optical table at the time. The best we can do is we have 3 microscopes side by side, and the best we can do is do 3 experiments at a time. So even though you can get, in the end, some nice migration movies of the cells, we really ask, is this really a lab on chip in such a small scale? Uh, Perhaps in reality, it's really a chip in a lab because there's so many other things that's also required. So that motivated us thinking about the possibility going from active to passive. What if you can eliminate the need of those external controls and still be able to perform cell migration assays? So this is one of the device we developed. So as you can see, one of the idea that people thinking to use passive microfluidic devices without using the pump is you putting some solution into a well, basically let the gravity to do the trick. And if that can maintain a stable flow, then it can basically have the same gradient generation that measure the migration from there. But the practical problem of this approach is when you are dealing with such a small volume of the chemicals, it's really difficult to accurately control the exact volume of the chemical into different wells. So only a very slight difference in the downstream channels is going to significantly shift the mixing interface. So our approach is to adding what we call the balance zone in there. So before the cell, the buffer and the chemoattractant were mixed in the downstream channels to generate the gradient, we would like to briefly meet first. They will allow them to compensate the difference of the pressure from their upstream wells. So eventually, when they melt in the downstream well, the pressure is balanced. So they will help to create a stable gradient just using the gravity pressures. So this help us to eliminate the requirement of the syringe pumps and tubings. The whole device can be made standalone. But the second issue that we are facing is if you load the cells initially everywhere in the channel, they will be subject to different gradient exposures, right? Because you have different concentration in the gradient. So the analysis of the cell migration later on can be complicated by this initial positioning of the cells. And the other related issue is they require lots of manual tracking to get the cell positions over the experimental period. If you think about you have 100 cells to get a good statistics, and you got 100 frames over an hour-long experiment, and you do the math. That's how many manual clicks you need to do after experiment to get enough coordinate for the calculation of migration parameters. So to address this issue, we propose what we call the docking device or the D chip in there. That is to make a very thin channel on one side of the device. So when the cell is first loaded to the device, it will not pass. So they are pre-aligned on one side of the device. Only when later you apply the chemical gradient, the stimulated of the cells were able to squeeze through this very thin roof into the gradient channel. This will give us the option to not track the cells and just measure the number of the cells eventually get into the gradient channel. If you want to go a little bit further, you can also further quantify the migration displacement of the cells into the gradient channel as a rough measurement of cell migration and chemotaxis. So one of the things we did using this D chip is to use that to study human neutrophil migration to the sputum samples of COPD patient. So as we know, COPD typically causing from smoking. Biologically, it is those inflammatory factors secreted by the airway induce the migration of neutrophils and potentially some other cell types too to the airway, which further damage the airway tissues. Traditionally, people use spirometry to diagnose COPD, which basically measures how fast and how much air the patient can breathe in and out. But we ask, is it possible that we can isolate a sample to a biomarker measurement and thus speak to using neutrophil chemotaxis as a cell functional biomarker? So we get a sputum sample from a COPD patient, and then we get a supernatant of the sputum and use that to generate a gradient. And then we load the human neutrophils into the device to measure their chemotaxis. And as we can see from here, they correlate quite nicely from this conventional spirometry measurement, even though the sample number from the patients is very limited, but it's rather a proof of concept demonstration. So the higher the cell migration responses, the lower the spirometry measurement from the traditional diagnosis method. And here just some images showing the migration assays using this D chip. So I won't get into details. FLP is working as a control, but you can see for COPD group, there's also lots of cells moving into the gradient channel. Okay, let me move to the next slides. Okay, so we found a way to get rid of the requirement of external pressure device and the tubing connections. We found a way to pre-align the cells at the beginning, so we don't have to track the cells, and also that will improve the accuracy of the cell analysis because all the cells has the same identical initial positions. But another issue also limits the efficiency of the migration assay is this lengthy cell isolation process. So typically you get the blood and usually it's a large amount and you apply the centrifugation to separate the cells into different layers. Then you extract the cell laser of the increase, put into buffer, wait a little bit, and that's how you can perform the migration experiment from there. We asked, is it possible that we can directly isolate the cells from a drop of blood on the same chip so we can perform the subsequent chemotaxis experiment all within the same platform? And this is what we developed, what we call the all-on-chip method. The idea is you take a small drop of whole blood from the donor, you mix it with the antibody cocktail, conjugate it with magnetic nanoparticles. Then you add the mixture into the cell inlet of the device. Then you apply a pair of magnet to trap all these magnetically labeled cells. And those are cells that we don't want, only allow the unlabeled cells negatively passing to the migration chip on the same device. And as mentioned earlier, we have this docking structure. So the selected cells is aligned, then we apply the gradient to induce chemotaxis. The whole process ideally can be done within 25 minutes, about 10 minutes for the cell purification on chip, followed by 15 minutes of chemotaxis experiment. So I show a quick movie to demonstrate the cell directly isolate from the blood on the chip using the magnet Then you align the cell initially on the top side of the channel. Then you apply a gradient, which is showing this fluorescent green color. Then you see this strong flux of cells migrating toward the chemical gradient. So this is further eliminating the need of a lengthy off-the-chip cell isolation process. Now, final thing, you still need a large specialized microscopy setup to perform the migration assay. So we finally asked, is it possible to develop an inexpensive and portable imaging device? So we can actually take this to different labs or even clinic and towards really help people to adapt the integrated microfluidic platform for cell migration evaluation. So very briefly, we developed an integrated, very small platform. So you have the LED light source. There's some lenses to help focus the light. Then you align this with a smartphone camera for imaging cell migration. And you can attach a very simple heater to maintain the temperature during cell migration experiment. And there's also a very small motorized stage, so you can do multiple position image acquisition. I'll show a brief theory of this critical imaging process. The device is, and then along with the smartphone camera, this is the actual movie taken by the smartphone camera. We call this the M kit, the migration kit. Everything can be put into a suitcase where you have the device, this imaging apparatus, use the reagent in there. We did a quick demonstration in a local hospital. This is in a doctor's office. We directly isolate the cells from the blood and perform the experiment. And here is some measurement in there. So for the medium control, you don't get many cells. And with the COPD gradient, you got a significant increase cell migration in this portable device. Okay, so I think I have probably 10 or 15 minutes. I'd like to continue this kind of a story. So we address the external pressure device issue, address the cell initial alignment issue, remove the requirement of off-chip cell tracking. Now we don't even need a microscope to do the migration assay. And then this all-on-chip method will also allow us to do on-chip cell isolation directly followed by cell migration assay. But one issue is still the throughput. If you can only do one experiment at a time, for something like human neutrophils, they don't stay alive very long after isolation. There's not many experiments that you can do. So that's the motivation for us to further improve the experimental throughput on the same device. Before we got the D chip, and then later we got the D3 chip, you can have 3 parallel devices running at the same time. We asked, can we further improve this throughput? So here is an illustration of what we call the DA chip. Now, it seems straightforward to move from D3 to DA or even higher throughput just by putting more and more parallel channels on the same device. But practically, there's a problem, and that is you don't want this chip to be too large. When it's too large, it makes it much harder to image. Even if you have this motorized microscope stage, if the distance between different imaging area of the device is too far away, it takes a long time to make the stage to move from one unit to another. So we have to find a way to make it more compact. So that's how we change this parallel design into this radio design. By doing this, all the imaging area of different unit can be concentrated in the center area of the device. That will make the imaging of all 8 units much easier. The related issue is using the balance zone D device I talked about earlier. because you need this additional compensation area to stabilize the pressure, you need a fairly long channel to also help make the flow more stable. And that's either for 3 parallel unit, but much harder for much more parallel unit, right? Because you have a lot of those zigzag channels that will make the total size of the chip much, much bigger. So to make a DA chip, we think about, is it possible to make just 3 channels without using the compensation zone? Now, if you don't have a compensation channel on the chip, how do you stabilize the pressure difference without using external pressure device? And this is where this oil coverage idea came along. And that is, we know we cannot practically control the pipetting of different volume at such small scale into different inlet wells. But what if we don't care about that? But after we adding the buffer and the chemoattractant, we cover both inlet with a common drop of oil. Because oil does not mix with buffer and the chemoattractant solutions, so we don't worry about contaminations. And if you pick the oil has a similar density of your reagent, now the pressure difference not only is subject to the volume difference between the 2 reagents, but it's further balanced by the density difference between your reagent and the oil. So that's briefly showing here in those equations. I won't get into details, but the idea is using this oil coverage, even the initial volume between the 2 inlets is fairly different, you will be able to get a balanced pressure to generate a stable concentration gradient. And here shows we intentionally pick 50 microliter for buffer and 45 microliter for the reagent. Without oil coverage, you can see the significant shifting of the mixing interface. With oil coverage, it goes back to the middle with a 50% divide. All right, so here is a demonstration using this high throughput docking device. And this is in collaboration with Dr. Thomas Clonisch's lab in Human Anatomy and Cell Sciences at the University of Manitoba. And he's particularly interested in a protein called HMGA2, who has been suggested to be involved in breast cancer cell migration and metastasis. So we wonder if we can use the microfluidic device to look at the effect of HMGA2 on breast cancer cell migration. So here we use this MDA MB231 cells, which is a human metastatic breast cancer cell line as a cell model for the experiment. And the movie showing here, those 231 cells squeeze over this very thin docking roof and migrate into the gradient channel in response to EGF gradient. Cancer cell migrate slow compared to neutrophils, and typically we require 3 to 6 hours for each experiment. If we have to do that one at a time, that's a lot of experiment, many experiment days. And here we're able to quickly get those responses to compare HMGA2 overexpressing cells and empty vector control for different conditions. So just very briefly, with our expression of HMGA2, the dose response is actually fairly similar to the empty vector control. So the idea is the endogenously expressed HMGA2 in cancer cells was sufficient to induce migratory responses. Given that additional HMGA2 does not significantly affect the migratory response of the cancer cells. And there we further evaluated the migration of the HMG2 knockout cells, the KO cells, use CRISPR gene editing, again, in collaboration with the Clonisch's lab, and also apply this inhibitor to block HMG2 functions. I won't get into details. The idea is using this GA chip, we'll be able to quickly evaluate different conditions only in a few weeks. So to get an idea of the effect of the knockout and also the inhibition effect. So as expected, both the knockout cells and the the cells treated with the what we call the Lin-20 inhibitor at 72 hours reduce significantly the migration of breast cancer cells in response to the EGF gradient. And in fact, the inhibitor treatment at 72 hours reduce cell migration even more compared to the HMGO2 knockout cells, which is kind of interesting to us, suggesting perhaps the inhibitor has a broader effect on cell migration as opposed to the specific inhibition of HMGA2 mediated cell migration. All right, so in the next 2-3 slides, I have my kind of last story to share with you today. That is not only this device can be used to evaluate single cell types, you can also use this to look at the interactions of different cell types. And here is example, we collaborate with Dr. Sam Kwan's lab in immunology of U of M, and this is mostly done by Jolly Hippolito, which is co-supervised by both Sam and myself in the lab at the time, and to look at the microarray interactions between natural killer cells and dendritic cells. And it's a fairly old review, but nicely demonstrate this bidirectional crosstalk between NK cells and dendritic cell in terms of their activation and maturation. So here we start by using the supernatant that we extracted from the dendritic cell culture upon LPS stimulation to generate a supernatant gradient in the microfluidic channel to mimic the NK-DC interactions, even though here we do not directly involve dendritic cells in the microfluidic channel, but use this supernatant as a mimic. So we saw this really strong chemotaxis of mouse NK cells towards the dendritic cell supernatant. So the gradient is from the left to the right, and in the control, not much of NK cell moving in, but a lot more cells moving in. We have a mature DC supernatant gradient.
I think we lost our speaker there. Give it a moment. I hope he comes back.
I'm a student and I'm just texting him to see if he's coming
Yeah,
back.
thank you. Worst case, if there's issues, maybe you could present your part. We could come back to Professor Lin after.
I think he's just reconnecting now.
Yeah.
Can you see me now?
Yeah, we could see you.
Okay. I don't know what happened earlier.
Maybe the storm.
Yeah. So let me share my screen again.
Yeah.
Yeah, I apologize for the disconnection earlier. I have to reconnect back, I think, twice. But I almost got to the end of my slides talking about this last example of NK-DC interaction. So we pattern dendritic cells in there and directly look at NK-DC contact times. Then we can have some quantification from there to compare NK interaction with immature DC and mature DC. So we call this chemotaxis facilitated NK-DC interaction from there. But one of the issue in there is the dendritic cell patterning in the gradient channel is not controlled, right? In some cases, you got more dendritic cells in there, and less cells in some other conditions. So it's hard to really quantitatively compare the result in there. What if the interaction is not really because the crosstalk between the two different cell types, but rather because this initial variation of dendritic cell patterning. So we further developed what we call the T2 chip. That is further configure many small, we call it conversation rooms on one side of the channel. So we limit the contact area for NK cells to talk to the DC. Then later on, we quantify the interaction in each small trap channels, then do the average over many of those small traps. So here is the example that we, for instance, label the pre-pattern dendritic cells. The NK cell move in, and so they talk to each other. We can quantify the contact time again. So we saw this further improve the accuracy and control of NK-DC interactions. And that is pretty much all the example I plan to share with you today. We are gradually moving toward making the cell migration assay more standalone, making more automated. So that's our vision toward automated, eventually hopefully run by itself cell migration assay, and potentially the same idea can be used for many other biomedical applications. So with that, I'd like to thank all my lab members, current and the past, all the collaborators, both local and in other places, and the support from funding agencies and the industry. And again, thank you for the listening, and put Nick's picture and a snapshot of his work in there that he's going to talk about his work using this new wireless non-contact electrofield apparatus to look at its effect on both immune and cancer cell migration. So thank you.
Thank you. Nick?
Yeah. All right. Perfect. Do
You're
you want
up.
me to? I'm up.
You're up.
I'm up. Awesome. Okay. All right.
Yeah, we'll save the questions at the end.
Sounds good. Can you see my screen?
Yes, we can see it now.
Awesome. Perfect. Alrighty. So yeah, as Dr. Lin said, my name is Nicholas. I'm a PhD student in physics in his lab, and I'll be presenting on the differential migratory responses of cancer cell and immune cell to wireless electrical stimulation. So first off, as Dr. Lin talked about, directed cell migration is when cells move in response to various types of environmental cues, such as chemical gradients or electric fields. And again, we can see on the left, we just have the chemotaxis of the neutrophils. And on the right, we can see this electrotaxis of these epithelial sheets. So why is this relevant though? Well, it's relevant for several different reasons. For wound healing, which we kind of talked about, embryonic development, metastasis of cancer, and of course the big one, immune cell trafficking. So if understanding these processes are leading to technological and medical advances in the health industry. So for electrotaxis here, we can see that on the left, we have these MDA MB231 breast cancer cells, and they want to migrate in the opposite direction of the electric field. And this is actually due to the fact that there is a potential difference between between the inside and outside of a normal breast duct. And these cells pick up on that potential difference, and they want to metastasize and go towards the normal breast duct. And the neutrophils, on the other hand, we can see on the right, they go towards the direction of the electric field as soon as the electric field is on. And again, this is for immune cell trafficking as well, similar to kind of what Dr. Lin talked about in chemotaxis. So the migration of cells was first due to an electric field was observed by Denier in 1892, and he termed the word glavoltactus. It's now more commonly known as electrotaxis. And we can see that there's different types of these electrotaxis assays. So we have on the left is the more traditional contact-based one. And this one, essentially, you have two agar salt bridges, and you directly put the electrodes with contact with the cell medium. And this is what more traditionally has been done. But then nowadays, we're actually moving on to more wireless approaches. So wirelessly, we can either have a transmembrane or transwell assay where we create a alternating electric field by making a changing magnetic field. Or again, you can also have this direct current non-contact system by applying a voltage difference between two copper plates. So our objectives are to build a more customizable device capable of both Petri dish and microfluidic assays under wireless direct current. And we want to use this direct current because it's more physiologically relevant. And we're going to test this device on single cell migration response of breast cancer and immune cells. So we can see that this is not the first time that this wireless non-contact direct current has been done. But from the very few literature that is out there, we can see that they gave clear collective cell migration responses, but no single cell migration responses. So we'll just note that collective cell migration was, again, like the epithelial sheet, you look at all the cells moving as a whole, whereas single cell migration is kind of more relevant to immune cells, where you're tracking each individual cell. So yeah, these literature found that there was no single cell migratory response due to this wireless direct current electric field, but they did get something with this cell or collective cell migration. So we kind of wanted to optimize this a bit and see, okay, can we actually reproduce this? And if not, can we actually get single cell migration due to this non-contact direct current electric field? So we did this by giving an entire overhaul to the design. We can note that our design is engineered in such a way that it can allow for not only classical Petri dish assays like the previous one done by a different group, but we can see that it also we can allow for microfluidic devices by enabling the use of these 3D printed cell chambers that will pop in and out of the apparatus. We also note that we developed the device in such a way that it's very user-friendly. So we have removable copper plates, and we can change the distance between the copper plates as well. And we can see this is an actual image of the 3D printed device, where we can see how it'll work with both microfluidics and also your traditional Petri dish assays. So another thing we changed was that the previous device had an electric field gradient, and they did this by having an aluminum ring around the actual Petri dish. So more traditional electrotaxis assays do not have this electric gradient. They have a relatively uniform electric field. And this is because, by definition, the electric field is actually a voltage gradient. So we decided that we wanted to have this this feature, and we also note that we are doing relatively high electric field strength. So we do 5 volts per centimeter and 10 volts per centimeter. And we do this as well because their electric field strength was relatively low. So now let's talk about the cell migration assays that were performed. So we did both human neutrophils and we did MDA MB231 breast cancer cells. So for the human neutrophils, cells were magnetically isolated from whole blood. We did coating of 100 micrograms per liter of collagen with 2% BSA blocking. We seeded cells for 30 minutes, or we seeded 500,000 cells, and our experimental time was 30 minutes. And to get cells to adhere to the substrate, we pre-stimulated them with 20 nanomolar of FMLP. And for the breast cancer cells, they were cultured in our DME F12 media with 10% fetal bovine serum and 1% antibiotic. And these cells were serum starved for 24 hours before the experiment, and we loaded 200,000 cells onto 20 micrograms per milliliter collagen 1 substrate blocked with 0.4% BSA. And we can kind of see in these figures the overall trajectory of our experiments. So we start with, our cell culture or substrate coating, and then we do our cell seeding, our setup, time-lapse microscopy, and then our data analysis. So for our data analysis, we use the manual plaque tracking plugin from ImageJ, and we created a custom Python script that'll analyze various quantitative cell migration parameters. So in particularly, we can see we have 3 layers of parameters. We have our motility layer, which has things like our displacement, our velocity. We also have our orientation parameter layer, which have our orientation index and a directionality component. So we define the orientation index to be the displacement over the total distance of the cell trajectory. And what this corresponds to is a orientation index close to 1 means that the displacement and distance is very close to each other. So that means the cell traveled in a linear path. Whereas if the cell made lots of turns back in on itself, or it didn't travel in a very linear path at all, it would be an orientation index close to 0. And then we note that the x directionality component is again just the cosine of theta, where theta is the angle between the final displacement vector and the initial starting position. And then we also quantify orientation index in the x direction, which we're just looking at the x component now over the total distance instead of the total displacement. So our results here, we can see that when tested with our breast cancer cells, we see that this wireless direct current electric field, oh, I don't know why they're not playing. Oh, there we go. The wireless direct current electric field, it reduces breast cancer cell migratory persistence. So on the left here, we actually see the trajectories of the cell under the real time-lapse imaging. And on the right here, we can see our normalized cell trajectory. So we basically put all the cells start at the origin and then track the trajectory. And this makes it visually where we can actually see the trajectories more qualitatively. We notice there's two different lines. So there's red lines and black lines. So the black lines represent cells that are traveling against the electric field, where the red lines are representing cells that travel towards the electric field. And we can see from that, along with these more trajectories here, that it's about 50-50. So half the cells are going towards the electric field, half the cells are not. And this is more quantified by the angular histogram in figure B here. So our motility analysis, when we analyze those parameters, we can see that there's an increase in velocity, but we also must note that this wasn't as reproducible over multiple experiments. But what was very reproducible over multiple experiments was this decrease in migratory persistence or orientation index. So we can see a statistically significant decrease in this orientation index with increasing electric field strengths. Now, for the immune cells, we have that this wireless direct current electric field, it induces cathode-based migration of neutrophils. We can kind of see that again in the plot on the right here. We see that there are more red lines and black lines, so it would appear that more neutrophils are going towards the electric field than against the electric field. And this can be further realized by looking at our plots for our distributions here. And we can also see this by our angular histogram. And when noticing the motility analysis, we can again see that there is an increase in the displacement and a little bit of increase in velocity, but this was not as reproducible. So we don't want to make any statements here. And we also notice for our orientation index, there was no statistical differences here, but we do see that there is a general trend for the x directionality and the orientation index in the x directionality. So we actually further did some correlation analysis of these neutrophil displacement, and it allowed for directionality of thresholding. So essentially what we have here is we did a correlation fold change, and we note that when comparing the correlation of orientation parameters and the displacement or the orientation parameters and the motility parameters, we see that there's a roughly 2 times increase in orientation or in correlation between those parameters. So we can see from figure C here, we have for the displacement parameter and the x directionality, it's about 1.9 times more correlated. And then in figure D, we can kind of see how this correlation tracks over time. So what this allows us and what this tells us is that the cells moving farther away from where they started tend to go in the direction of the electric field. So by using this, we decided to do a displacement threshold. So we took the top 25th percentile of cells under how far they traveled away from the origin. And we can, when doing this analysis, we can see that it is much more clear that out of these top 25th percentile of cells, that most of them go towards the electric field. And so now we can see that our orientation analysis, we have some statistical differences, and we can also note the relative y-axis here. So before it was about 0.2 was the max. Now it's shooting up to about 0.3 or 0.4. So you can see it nearly doubled the orientation parameters. So finally, we have our conclusions and future work here. So we demonstrated the effective and flexible use of the wireless direct current electric field device for cell migratory studies. And this provided the first ever evidence of single cell migratory response of immune cell and cancer cells due to this non-contact direct current electric field. And then we also note that we got a differential response of cancer cells and immune cells, which suggests possible underlying cellular mechanics. So from there, we'll just recap. We have our neutrophils. We found that under this wireless electric field, they tend to go towards the electric field. And whereas cancer cells under this wireless electric field, they tend to not go towards the electric field, but they tend to lower their migrating persistence, which means they essentially make more turns. So for future work, well, we want to test this with microfluidics. Now that we know that this system is working and we found some evidence that this non-contact direct current electric field actually does induce cell migration, we want to test on microfluidics to get more physiologically relevant situations. And then we can also do this where we can add chemical gradients as well, so we can have an opposing chemical gradient. And yeah, that is it. So my acknowledgements are here. And yeah, thank you.
Thank you. Actually, that was quite incredible. Both talks were very much amazing. And especially Professor Lin, I really like the microfluidic devices. That was just incredible what you came up with. So before I open up the floor for questions, I have a couple questions myself. So I was wondering, for the first part, Professor Lin, you presented some work at the end with dendritic cell neutrophil, I think, co-culture experiments, if I'm not mistaken. Yeah, I was wondering if you're considering looking at cancer metastasis, maybe, and looking at two different cell types to see how that could, whether there's any regulation between migration in your device there.
Yeah. Indeed, we're actually very interested in looking at immune cancer interactions using those devices. We have found similar to the NK DC interactions that I presented in there. When we take the supernatant from a breast cancer cell line culture, for example, and generate a gradient in the device, we see immune cells has shown some quite interesting responses. Neutrophil, for example, has a strong chemotaxis towards the supernatant gradient from the cancer cell culture medium. T cells, interestingly, in some cases that we observed, they were first migrate towards the supernatant gradient, but at some point they got repelled away against the opposite direction. So that's kind of ongoing. And then we also start trying putting cancer cells into the gradient channel, and then to look at the physical contact between immune cells and cancer cells there as well.
It's amazing, yeah. That's cool. I had to see the T cells stop migrating at some point. That'd be interesting to determine the mechanism behind that and if that can be regulated. That's really amazing. And for Nick, actually, yeah, very incredible. I like that electric field work, especially like the idea of maybe putting an electric field, since the cancer cells didn't really migrate much, it was mostly the immune cells, right? You could put like an electric field around a tumor, let's say a solid tumor, and maybe hopefully attract, right, immune cells to the site, right, for let's say better treatment option or immune therapy of some sort. That's sounds like a pretty cool idea. And the other thing, the question I would have for you is, so I noticed that some cells were migrating more than others, right? You had a population of cells that really migrated quite a long distance versus the rest. Do you think that there's maybe some difference in those cell types, that there's maybe a mechanism or so, or something that's allowing them to migrate further in the electric field versus the other population?
Yeah, so good question. So again, there's similar things with chemotaxis as well, where different cell phenotypes will essentially migrate. Some will migrate better and worse. So actually, in one of Dr. Lin's experiments that he showed, that they kind of isolated the different distances, sort of. So they took the different distances of the cells. So for cells that migrated further, they kind of collected those ones, and they also analyzed cells that migrated less. In terms of the electrotaxis response here, it is very interesting because in traditional electrotaxis, like you put the two electrodes directly in the medium, the amount of cells that would migrate directionally would be about like 95%. Whereas in this wireless case, we're not seeing that. Even with the neutrophils, we are seeing that there is some directional migration, but it's not to the level that would be expected in our traditional contact-based. And we are hypothesizing actually that this may be due to the lack of current in our system. So electrotaxis is one of those things that the underlying mechanisms is not necessarily really understood as well as chemotaxis. And in all previous electrotaxis experiments, when you have cell culture or when you have cells or electrodes directly in the medium, that when you apply a voltage difference, it creates an intrinsic current. Which also induces other things like pH changes and stuff like that. In our system, we don't have that. So we're hypothesizing that maybe the directional response is more towards current instead of electric field. And that also might explain why our cancer cells didn't directionally migrate, whereas traditionally they should have.
But you did have like your neutrophils, right? Or the migrate, right? You
Yes.
had, yeah. To exploit that in some way would be pretty interesting for like immunotherapy. Maybe
Exactly. And on that, especially with what you mentioned about the whole tumor idea, exploiting this differential migratory response could be very useful in controlling or sending immune cells to a tumor while keeping the tumor not metastasizing.
Exactly.
Yeah, so it
Exactly.
has very high potential for biomedical applications.
And the strength of the field, does that have any? I'm not sure. I'm not sure, or I don't remember if you looked at that. But if you regulate the strength in some way, does that have any effect?
Yeah, so I didn't show it here, but for lower strengths, there was actually no cell migratory responses. So it's only when we're increasing the strengths here to what's more physiologically relevant and even increase in what is physiologically relevant, we're actually starting to see this migratory behavior. So again, this suggests that the underlying mechanisms for maybe wireless non-contact electric fields is a lot different than perhaps with the direct current one, where it'd be more physiologically relevant, so a little bit lower electric field strengths.
Perfect. Well, amazing. Thank you for that presentation.
Thank you.
I'm not sure if anybody who has any questions, please feel free to unmute. You can also write in the chat. Well, there are no more questions. Thank you very much, both of you, for your presentations. That was great, fantastic. And good luck on your the future work. I'm sure it's going to go quite far. You can see the potential there.
All right. Thank you very much to everyone for inviting me and my students. Great pleasure for them today.
Thank you. And goodbye, everybody.
All
Thank
right.
you.
Bye-bye.